All right, so now we are at chapter 6. So we've looked at the parts of cells, we've talked about chemistry. Now we're sort of seeing some of the active chemistry that goes on in cells. And so in the why cellular respiration matters, they give you a couple of examples. One being metabolic processes, right? So metabolic processes, meta, change, so processes that, in this case, we talk about breaking things down. We're breaking down, typically talk about glucose into byproducts. Well, the process that produces acid in your muscles after a hard workout are similar to the processes that produce things like pepperoni and soy sauce, yogurt, and even bread. The cells in your brain have a high metabolic rate. They go through a lot of energy. The cells of your brain burn through about a quarter pound of glucose every single day. Also, you have something in common with a sports car. Both of you require an air intake system to be able to burn fuel efficiently, right? So needing oxygen as part of the process. Now, for many endurance athletes, the rate at which oxygen is provided to working muscles is the limiting factor in their performance. Right? The ability to pull in enough oxygen to keep the reaction of breaking down glucose to provide ATP. That's the limiting factor. If you had an endless oxygen supply, a person could be active much longer. So your aerobic capacity is the maximum rate at which oxygen can be taken in and used by your muscle cells, and therefore the most strenuous exercise your body can maintain aerobically. All right, so aerobic means with oxygen. So when you watch some of these athletes, you think about it, one of the reasons you train is not just to build up muscle or build up, increase your coordination, but also to, in essence, learn, get your body better at gas exchange. Now, if you work hard and exceed your aerobic capacity, I usually joke about this as the running for the bus moment for most of us, where you're running, and eventually, if the bus is mean and doesn't stop for you, you get to the point where the demand for oxygen in your muscles will exceed your body's ability to deliver it, right? So now, if you don't have oxygen, you can't, your aerobic metabolism can't keep going. The metabolism then switches to anaerobic, and meaning without. And so it's basically an emergency mode in which they are still producing energy, but it's really inefficient. And instead of breaking that glucose all the way down to water, carbon dioxide, and ATP, you wind up breaking it down to breaking it down to lactic acid, which is a big byproduct. So you haven't broken down all those chemical bonds and converted that energy in those chemical bonds to ATP. And the old expression, feel the burn, refers to people exercising past the point of aerobic capacity, and eventually the lactic acid causes discomfort in the muscles. Okay, so energy flow and chemical cycling, right? We're back to this again. So all life requires energy, and as I said in the previous section, energy enters living systems and eventually exits in the form of heat. Now, in almost all ecosystems on Earth, this energy originates from the sun, the exception being things like those deep sea vents, like I talked about in the last section. During photosynthesis, photolight synthesis plants are converting the energy of sunlight to the chemical energy of sugars and other organic molecules. So using light, they're synthesizing sugars. Animals don't do this, right? We're animals. We can't, unfortunately, lie out in the sun and get a good meal that way. So all things that are heterotrophs, animals like us, depend on that conversion for food and more. So we either directly eat the photosynthetic organisms or we eat things that have eaten photosynthetic organisms. So plants and other autotrophs, auto meaning self, troph referring to feeding, these are organisms that make all their own organic matter from nutrients that are entirely inorganic. So if you think about this, if you have a plant, right, you water it, It gets its carbon dioxide from the air, and you might have to supplement the soil with minerals, but it's making all its organic material, all the cellulose, the starches, the sugars, all that from totally inorganic substances. So autotrophs are self-feeders. Basically, in the context here, it's sort of equivalent with photosynthetic organisms, because there are chemosynthetics, but those are the guys down in the deep sea, and we sort of ignore them at this point in the course. So autotrophs in this context are pretty much all photosynthetic, photoautotrophs. Heterotrophs, hetero meaning other, these are other feeders. They have to eat other things. So humans and other animals, also fungi. These organisms can't make organic molecules from inorganic ones, right? You have to eat your organic molecules. You can't make them on your own. And most ecosystems depend entirely on photosynthesis for food. Again, exceptions being like the deep sea vent bacteria. Biologists refer to things that are producing food, right? Plants and other autotrophs as producers, right? They're making the food. They're producing the food. Things that have to eat something else, are the heterotrophs. We call them consumers because they have to obtain their food by eating plants or eating things that have eaten plants. For example, the koala. This is a guy that, believe it or not, they actually carry out a type of fermentation in their stomachs. They eat this very hard to digest food called eucalyptus. And they basically, if you see them, they sort of sit back and they look almost like a little Buddha figure. Well, that big belly they need a lot of real estate because they have to process a lot of plants to get enough nutrients out of it. Now, I talked about energy flow back in chapter 5. Now we're back talking about chemical cycling between photosynthesis and cellular respiration. The chemical ingredients, right, the reactants for photosynthesis are carbon dioxide and water. Carbon dioxide passes from the air into a plant via tiny pores. So there's little pores in places like the leaves. So in that photosynthesis experiment, one of the things you have to do is you have to pull the gas out of those leaves and replace it with water. What's in the water? Bicarbonate. This is going to be the source of carbon dioxide for those plants in those discs in that solution to carry out photosynthesis. The water is, in regular terrestrial plants, absorbed from the soil by the plant's roots. Inside leaf cells, chloroplasts use that light energy coming in to rearrange the atoms of carbon dioxide and water to produce sugars. Most importantly, glucose, C6H12O6, right? So 6 carbons, 12 hydrogens, 6 oxygens, and other organic molecules. The byproduct is oxygen gas, right? So oxygen is a waste gas in photosynthesis. And this is sort of showing you the movement of energy and chemicals in ecosystems. So if you start with the sun up at basically 12 noon in this, if you think of this as a clock, sunlight energy enters the ecosystem, right? There's those chloroplasts in the cells of photosynthetic organisms, traps that sunlight, and that energy is used to break and rearrange bonds in carbon dioxide and water to yield, now we go to 3 o'clock on the clock face, glucose and oxygen. Well, what's necessary for respiration? Glucose and oxygen. So the stuff that was produced in the chloroplasts of an autotroph is now going to be consumed in the mitochondria of both autotrophs and heterotrophs, right? So both use that glucose for energy. It's just that one makes it itself and the other has to consume it, has to ingest it. A product of that process in cellular respiration, which we're about to go into in great detail, is ATP, right? So some of that energy from breaking apart that glucose molecule is going to be converted into the chemical form of energy ATP, and that's what drives cellular work. You also have heat energy, because no energy conversion is perfect. You're going to have some heat energy exiting the system every time you're breaking down that glucose. That's why animals like us are warm, we have a high metabolic rate. We have a high rate of burning through glucose molecules. Well, one of the byproducts of that is warmth. All right, so products of cellular respiration along with ATP is water and carbon dioxide, which is, of course, what the plant needs to carry out photosynthesis along with the sun energy. So each depends on the other. Plants need us to produce carbon dioxide and water that's released to the environment, and we depend on the autotrophs for glucose and taking up oxygen in the environment. Also, something to keep in mind, one of the concerns with climate change is buildup of carbon dioxide in the atmosphere. So one way people are trying to address climate change is to just plant more plants, right? Because plants are carbon dioxide sinks. If the plant can absorb carbon dioxide, tie it up in their cellulose and their sugars, you're pulling it out of the atmosphere. So some of the solutions for addressing climate change include increasing the amount of green stuff and hopefully stopping people from burning down lots and lots of trees, which were pulling up carbon dioxide out of the air. All right, so cellular respiration uses oxygen to convert the energy stored in the chemical bonds of sugar to another source of chemical energy called adenosine triphosphate. So Mary talked about cashing a check, right? So you've got that energy in the chemical bonds of sugars, right? So that would be the check. You have to convert it to you have to convert your check. You have to cash in your check to be able to use that value in a store. Well, you cash in a sugar check to get adenosine triphosphate cash. That's the energy you can directly use to get stuff done in a cell. So in plants and animals, the production of ATP during cellular respiration occurs mainly in mitochondria. The waste products of cellular respiration, again, are carbon dioxide and water, which are the same ingredients used for photosynthesis. Now, plants usually make more organic molecules than they need for fuel. That surplus either produces material for the plant to grow, like cellulose, right, making more cell walls and producing more cells, or can get stored as starch in potatoes, for an example. So one way we store excess energy is as fat. These guys store excess energy as starches. I'm just going to pop over these. All right. So cellular respiration, most of us think respiration, we think breathing, right? Respiratory system. Cellular respiration is aerobic harvesting of chemical energy from organic fuel molecules. This is the main way in which chemical energy is harvested from food, either self-generated like an autotroph or consumed like a heterotroph. That chemical energy is harvested from food and converted to ATP. And again, aerobic means it requires oxygen. Cellular respiration, therefore, requires that a cell is going to exchange two gases with its environment. One, taking in oxygen in the form of O2, and getting rid of waste in the form of CO2. So that's what I mean by respiration, right? You've got to be exchanging these gases with the surroundings. So if you look at this illustration of a person exercising, right? They're breathing in and out. Well, the lungs are extracting oxygen from the air, and they're giving off carbon dioxide to be expelled from the body. So taking out the human part here. So again, carbon dioxide out, oxygen in. When that oxygen comes in, it's being what? Delivered to cells with mitochondria that are breaking down glucose. So that oxygen is used to as part of cellular respiration. That waste product, carbon dioxide, diffuses into the blood and gets brought to the lungs to be exited from the body. So that's why we talk about the respiratory system, but keep in mind, if you hear me say cellular respiration, I'm actually referring to this process in mitochondria. And one of those characteristics of life from the first section is transformation of energy and matter, right? All living things have to use energy. They have to manipulate, they have to change matter. So cellular respiration is a whole bunch of chemical steps, each with a specific enzyme to catalyze each reaction. This constitutes one of the most important metabolic pathways for nearly every eukaryotic cell and provides the energy these cells need to maintain the functions of life. So in the next illustration, we see this in the most simplified way possible. So again, the raw material of now this is a balanced equation. If you haven't had chemistry, the general equation is C6 plus O2, etc. But to have a balanced equation, you have to account for everybody, every atom that's involved in the reaction. So to make this a balanced equation, we don't just say, sugar and oxygen. Specifically, it's again, we typically talk about glucose. It's this sugar molecule, in this case glucose, plus 6 O2 molecules, right? So a total of 6 times 2, 12 oxygen atoms in the form of gas. There's also 6 oxygen atoms from the sugar. There's 12 hydrogen atoms, and there's 6 carbon atoms. So those are all the things going in. So what's on the other side of the reaction has to match that. You have to the same amount coming out as going in, because you never create or generate matter. You just rearrange everything. All right, so in a balanced equation, glucose plus 6 O2 molecules yields 6 CO2 molecules plus 6 water molecules and about 32 ATP. So let's count it up. We've got 6 carbon dioxide here. All right, there's the 6 carbon dioxide there. You've got 12 hydrogens here. Okay, 6 times 2, there's 12 hydrogens there. We have 6 oxygen here plus 12 oxygen, so that's 18 oxygen. All right, 6 times 2 is 12, plus 6 is 18, right? So you've accounted for all the atoms coming in and going out. And of course the ATP, which you don't, it's not one of the rearranged things. It's just that energy is used to make ATP. So that's why it's not one of the reactants or products in the same way as these other 4 materials. Now, the main reactions that make up cellular respiration can get grouped into 3 main stages: glycolysis, citric acid cycle, and electron transport. And that first step, if you think about glycolysis, right, you're lysing, you're splitting sugar. The second one is citric acid cycle. So this is something where there's an element that's going to not just go in and exit as a product. There's parts that are going to be cycling in it. So you'll see something where reactions yield products that go back into the cycle. And electron transport. And electron transport is going to be where you actually generate the most ATP. You're going to get a little bit in glycolysis, a little bit in citric acid, but what you're doing in these steps is breaking down that glucose. And in the process of doing that, you're releasing energy. And that energy is being captured in the form of high energy electrons, which eventually all get brought to this last stage. And those electrons are going to move through a series of reactions. And this is where most of your ATP is made. So again, keep in mind that both heterotrophs and autotrophs have mitochondria. So this is not something where one gets and one has it and one does not. So if we were to look in the mitochondria that you'd find in either of these cells, okay, here's that membrane-bound organelle. So it has this outer membrane and this highly folded inner membrane. So if you think about it, if you see a lot of folds, that means what? That's where the reaction's happening. To have more reactions take place, you could either have a really huge mitochondrion or a smaller one with lots and lots and lots of folds, increasing the surface area where that reaction takes place. So in the cytoplasm, right, this is the cytoplasm surrounding all the organelles, you have the first step take place. So the first stage happens in the cytoplasm, not inside the mitochondria. In that first stage, you have the glycolysis. You split glucose into 2 pyruvic acid molecules, and you generate a net couple of ATP. Those pyruvic acid molecules then enter the mitochondria. Everything from here on in requires oxygen. This stage does not. So out in the cytoplasm, you don't need oxygen. Once you get in the mitochondria, you need oxygen. So those two pyruvic acids enter the mitochondrion. They go through the citric acid cycle, and so you go from one molecule broken into two smaller molecules, and here you finish breaking them all the way down. So you no longer have, you basically just totally disassembled that glucose. And along the way, you've been capturing the energy that was holding it together. And here you directly make a little ATP, but most of the energy is being carried by things that captured those electrons when you broke down those molecules. All that energy gets brought to this electron transport chain, in which those electrons get used to make a lot of ATP. So most of the ATP is generated by this last step of the process. Okay, so we're going to go through each of these in detail, and at the end, hopefully it'll tie together. So, starting with glycolysis. During glycolysis, a molecule of glucose is split into two molecules of a compound called pyruvic acid. And this is taking place in the cytoplasm. And it also does not require oxygen. So when we get to fermentation later, we're actually going to be looking at variations on glycolysis. The next stage, the citric acid cycle, sometimes in other texts, or especially older texts, you might see it called the Krebs cycle. It was named after a guy named Krebs who determined how this thing worked. They're trying to sort of get away from naming things after people, so they're interchangeable. It's just you're going to see more and more citric acid cycle versus Krebs cycle. So in citric acid cycle, you finish breaking down the glucose, right? You split it in half in glycolysis. Now you finish breaking those two molecules all the way down to carbon dioxide. That, of course, is your waste product, right? Carbon dioxide being one of the waste products of cellular respiration. The enzymes for that citric acid cycle are dissolved in the fluid within mitochondria. So that breakdown is in the fluid stuff on the inside. I don't know if we talk about stroma and anyway, the fluid inside the mitochondria. So glycolysis and citric acid cycle generate a little ATP directly and much more ATP indirectly, because when those molecules were being broken down, that energy is being released, and some of it's being captured in the form of higher energy electrons, and they're being basically carried away by these electron carriers. The primary one is NAD+, right? So NAD+, nicotinamide adenine dinucleotide, that acquires the electron, right? So if it's got a positive charge, you add the electron, and now it's NADH. It's now lost the charge, right? Because that electron has balanced that positive charge out, so now it's a neutral. And a hydrogen proton is bound to transfer that energy, right? So you use the energy to stick a hydrogen onto NAD+, and you get NADH. Now you have loaded up an electron carrier. This is the molecule that's going to act as a shuttle, carrying high energy electrons from one part of the cell to the other. This is the energy that's going to be used to stick that phosphate, that third phosphate, onto ADP. Now, most of that's going to happen at the end. Now, the third stage, where the bulk of ATP production is electron transport. Electrons captured from food by NADH are stripped of their energy a little bit at a time. So it's like a bucket brigade, and you're pouring. People have in a line have buckets, and you're pouring water from one person's bucket to the next. And every time you do, you lose a little bit of water, right? So think of it this way. So as the water's being passed along, that some of that is being used to make something happen. As the electrons are being passed along, they're giving up a little energy each time, and that energy is being used to make a gradient, which I'll go into in more detail later. It's being used to set up a reaction that will be the stage that is going to be attaching that third phosphate to ADP. what do you do with those used up electrons in essence at the end? They get hooked up with oxygen to form water. So that hydrogen that you see in the NADH is going to bind with oxygen, and now you are going to do is get those electrons bound up in a safe form, right? Water is a very safe compound inside cells. Electron transport chains are embedded in the inner membrane of the mitochondria, right? So that's where all those folds were. Electron transport from NADH to oxygen releases the energy your cells use to make most of the ATP. So this is the way in which you take the energy that was holding together glucose and use it to make ATP. So this whole process is taking energy from one chemical source and converting it into another chemical source. So that overall equation shows that the atoms of the reactant molecules are rearranged to form the products CO2 and H2O, like I showed you in that previous graphic. So all you're doing is rearranging molecules. You're not making any, you're not just destroying any, you're just rearranging stuff, and that's what chemical reactions are. It's just rearranging stuff. The main function of cellular respiration is to generate ATP for cellular work. This process can produce about 32 ATP molecules for each glucose molecule consumed. Now, depending on which book you're looking at, one textbook over another, you may see 32, you may see 34, you may see 36. It's because it's never perfectly efficient. So just keep in mind that you may see other 30-odd numbers representing the amount of ATP generated. I won't make you pick between one and the other. So if you see a value on a test like, how many ATP, if you see 32, okay, that versus say 2 or 4 or whatever. So just keep in mind, you may see other numbers if you're looking at different sources of information about respiration. So here's a video.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy in fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, one carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
Okay, so we're going to go through the process step by step. We're going to start again with glycolysis. This again takes place in the cytoplasm outside of the mitochondrion and doesn't require oxygen. When you watch these reactions, you don't see oxygen going into this. So keep in mind that this one step can be done anaerobically. So during glycolysis, a 6-carbon glucose molecule is split in half to form 2 molecules of pyruvic acid, right? So you're going to go from one big 6-carbon molecule to 2 3-carbon molecules. That first split requires an energy investment, right? So there's the old expression, you have to spend money to make money. You have to invest energy to break down this molecule, right? So you have to spend 2 ATP molecules for glucose to get this going. The 3-carbon molecules then donate high energy electrons to NAD+, forming NADH. It also generates 4 ATP molecules. So if you spend 2 and you make 4, you have a net gain of 2 molecules of ATP per molecule of glucose. So again, following this in the form of a diagram. So there's your 6-carbon glucose in the beginning. Okay, P's represent phosphate groups. The little electrons are the little minus signs in the little spheres. So glucose is, you're going to invest 2 ATP, so you're converting ATP to ADP. You've transferred that phosphate group, and now you have these 2 molecules. So that you've got here's 3 carbons and then 3 carbons from the original glucose. You've added on these 2 phosphates because you had to expend energy to get this going. Next you're going to do is you're going to add on another phosphate. You're going to convert some energy and hydrogen protons to form NADH. And now in the next step, and again, this is happening for each of the 2 molecules, 2 new 3-carbon molecules, you then do is you're going to transfer those phosphates to 2 ADP, forming 2 ATP. So you spent 2, you made 2 from one, 2 from the other, so you made 4. So you have a net of gaining 2 ATP. So once you've done this, the thing that's left at the end, the product, is 2 pyruvic acid molecules, right? So what goes in? 6-carbon glucose. What leaves, what's the end product, is 2 pyruvic acid molecules and 2 ATP and a couple of electron carriers loaded up. All right? So the splitting, you've spent 2 ATP, so that phosphate from the adenosine triphosphate is transferred to each of those 2 3-carbon molecules. So you've expended 2 ATP. You then, and I try and keep it sort of simple. Keep in mind that there's like enzymes in each of these reactions and stuff. So believe it or not, it doesn't look it, but I'm trying to simplify this. Basically, you attach another phosphate to the three carbon atoms, three carbon molecules, and you strip off an electron and transfer that to NAD plus to form NADH. And again, you do this for each of those 2 3-carbon molecules. So now you've got a 3-carbon molecule with 2 phosphates on each one. Next, you're going to do is transfer those phosphates to ADP to form 2 ATP per 3-carbon molecule. So you spent 2, you made 2 for each of those molecules, so you have a net gain of 4 ATP. Now, before pyruvic acid can get used by the citric acid cycle, it's got to be converted to a form the citric acid cycle can actually use. So it can't directly use that molecule. Each pyruvic acid is going to undergo a couple of changes on the way in. Each one is going to lose a carbon as carbon dioxide, right? So they're going to each go from a 3-carbon molecule to a 2-carbon molecule. That 3rd carbon exits as carbon dioxide, right? So it binds with oxygen and off it goes. The remaining fuel molecules with only 2 carbons left are called acetic acid. Electrons then get stripped from those molecules and transferred to NAD+, forming more NADH. And then finally, this is now going into citric acid. So this isn't citric acid yet. This is just converting pyruvic acid to a form that can go into citric acid cycle. Finally, each acetic acid is attached to coenzyme A, abbreviated as CoA to form acetyl-CoA. All right, so it's kind of like you dump one of the carbons and then you put on a hat on your way in. So looking at this in more detail. So input, pyruvic acid. Again, this is just the step between glycolysis and citric acid cycle. The pyruvic acid loses a carbon as carbon dioxide, right? So there's some of that waste gas. The acetic acid is then going to have electrons stripped off. You're loading up another electron carrier. And then you attach coenzyme A to that. And now what actually enters citric acid cycle is this acetyl-CoA. Right, so what goes in from glycolysis? Pyruvic acid. The output that's going to enter the citric acid cycle is acetyl-CoA. So this is the link between glycolysis and the citric acid cycle. Right, so again, you're generating carbon dioxide, right? So a total of 2 per glucose molecule. One thing to keep in mind is from this point through the next illustrations, you're typically following just one of the 2 halves of that original glucose molecule. So you're tracing one, but keep in mind that everything's going to happen times 2. And I'll hit this again when we get to some of the later diagrams. But just sort of a warning that we're sort of just following 1/2 of that original molecule at this point. All right, so pyruvic acid loses that carbon to carbon dioxide. You then have acetic acid, loads up another electron carrier, attaches to coenzyme A, and now we begin the citric acid cycle. This is now what enters this series of changes in the fluid inside the mitochondria. The citric acid cycle finishes extracting the energy of sugar by dismantling those acetic acid molecules, those 2-carbon molecules, all the way down to carbon dioxide, right? So those 2 carbon molecules, each of those carbons is going to bind to oxygen and leave as carbon dioxide. So now you have gone from a 6-carbon molecule to 2 3-carbon molecules to 2 2-carbon molecules, and eventually you even break those last 2 guys down so that all the carbons exit as carbon dioxide. To do this, acetic acid joins a 4-carbon acceptor molecule to form a 6-carbon product called citric acid. That's where the cycle is named from. So that 4-carbon molecule, it's going to do is pick up acetic acid, basically carry it through breaking it down, and then go pick up another acetic acid. And this is why they call it a cycle. That 4-carbon acceptor molecule just goes round and round and round, carrying acetic acid to get broken down. I used to call it kind of like a merry-go-round, or a kid hops on the merry-go-round and rides the horse around. Except this would be one where the kid got cut in half, so it kind of gets morbid. But it's the same sort of thing where the carrier just keeps going around and around and around, picking up acetic acid and, having it be broken the rest of the way down. So for each acetic acid molecule, because keep in mind, 2 are going to enter this cycle for each original glucose molecule. For each acetic acid molecule that enters the cycle as fuel, 2 carbon dioxide molecules eventually exit as a waste product. So each half lost 1 going in, each half loses 2 more. So each half of that original glucose molecule, right, is going to be what? 3 carbon dioxides. How many carbons are in the glucose molecule? 6. Well, because you broke that glucose down into 2 halves. Each half goes through these stages. So each half is going to be broken down into 3 carbon dioxide for a total of 6 carbon dioxide per glucose molecule. Right, so here's a diagram of this. So going in, you've got the acetic acid, you have adenosine diphosphate and a phosphate, 3 NAD+, and now a new electron carrier, FAD. So at this point, the citric acid hops onto the acceptor molecule to form a 6-carbon citric acid molecule. Right? So the acceptor molecule is just going to go round and around it. It's going to pick up that acetic acid and carry it through a series of reactions in which those 2 carbons are split apart and lost as carbon dioxide. And then the acceptor just goes around to pick up the next acetic acid. And that combination of the acceptor molecule plus acetic acid is citric acid, hence the name. So the output is for each half, right? So for each acetic acid, and there's going to be 2 per glucose molecule, you're going to get 2 CO2, 1 ATP, 3 NADH, and 1 FADH2. So for each glucose molecule, each original molecule, it's going to be a total of 4 CO2, 2 ATP, 6 NADH, and 2 FADH2. That's the total output per glucose molecule. Because at this point, we're just looking at what happens to 1/2 of that original glucose molecule. So as you saw in the diagram, some of that energy is used to produce ATP directly, right? If you go back to the diagram, right, you've got 2 ATP per glucose molecule. But much more energy gets captured in the form of NADH and a second similar electron carrier called FADH2. So loaded up, NAD plus is NADH, loaded up FAD is FADH2. All the carbon atoms that entered the cycle as fuel are now accounted for as carbon dioxide exhaust, and that 4-carbon acceptor molecule is recycled, right? The merry-go-round horse goes back to pick up the next passenger. So what do we do with all these electron carriers? They come along to electron transport. So during cellular respiration, the electrons gathered from food molecules eventually fall, losing energy with each step. Electrons are, if you think, one way of thinking about this is like I don't know if they still have slinkies. There used to be a toy called a slinky. It looked like a big spring. You'd put it at the top of the staircase, and it would basically step onto a step, and then it would sort of loop and step and loop and step. So it would look like it was walking down the staircase. Well, think about potential energy, right? There's more potential energy at the top of the staircase than there is at the bottom. So each time it went down a step, it loses a little bit of that energy. It's basically giving off a little bit of that energy in the form of kinetic energy. It's kind of like that. The electrons are going to get passed along a series of proteins, and each time they do, they lose a little energy as they do so. But that energy is going to be used to create an interesting gradient. So during respiration, ultimately, electrons are transferred from glucose in food to NAD plus or FAD to FADH2. Okay, so you have these 2 electron carriers. The primary one is NAD. That's why they sort of ignore FADH2. So most of the way you see NAD plus converted to NADH, Then that NADH is going to release 2 electrons into this electron transport chain. So from all those previous reactions in glycolysis and citric acid, all those electron carriers come and bring their electrons to this last series of reactions. And this is the electron transport chain, and these are the proteins that were embedded in the membrane that you saw, the inner membrane that you saw in the video. And again, with each exchange, that electron gives up a little bit of energy. So another way of looking at it, if you think of the glucose as the guy with the hammer at the bottom left side of this cartoon, right? It's giving up that higher energy electron. The electron is captured by an electron carrier, that's the woman up at the top, and then it's going to do is toss that electron into that little spinner wheel going downhill. So as it goes downhill, it's losing energy. That energy is being used to stick a third phosphate group onto ADP, forming ATP. So the overall effect of this is a transfer of electrons. During respiration, it's a downward trip, right? So the highest amount of energy is in glucose. Again, you can't really, every energy conversion is not perfect. So it's not like it can hand, that amount of energy directly all the way to the electron transport chain. So it's going to lose a little bit each time you do this. So transfer electrons from glucose to NADH to an electron transport chain and ultimately to oxygen. So your first electron acceptor is NADH, your last electron acceptor is oxygen. Here's another way of looking at it. So electrons from food are handed over to NAD plus to form NADH. Ultimately, they're going to do is give up those electrons at electron transport chain. And it's going to go through a series, again, like walking down the stairs. It's going to go through a series of steps, each time releasing a little energy. That energy is ultimately going to be used to generate ATP. So what happens with those poor little electrons at the end of the line? 2 electrons plus 1/2 O2 molecule plus 2 hydrogens yields what? H2O. So you mop up some hydrogen protons, and I'll tell you why that's important in a minute. You get rid of those spent electrons, and you form a very safe waste product. And that's why anything that interferes with oxygen is going to be a limiting factor for generating energy, right? At the end, you have to have something grab up those spent electrons and soak up some of those hydrogen protons. That's the oxygen's role. If you take oxygen out, this whole thing grinds to a halt. I'll show why in more detail in a moment. So, the molecules of electron transport chains are built in the inner membranes of the mitochondria. So, citric acid cycle occurred in the fluid in the mitochondria. These reactions occur in the inner membranes, the proteins built into the inner membranes of the mitochondria. Because these membranes are highly folded, that big surface area can accommodate thousands of copies of the electron transport chain. So again, the whole structure and function, if you have a reaction taking place on a surface or absorption taking place on a surface, the more surface you have, the more efficient it works. Now, the energy stored by electron transport acts something like water behind a dam. If you think about a hydroelectric dam, right, you put a wall, say in the middle of a river, and that water piles up behind that wall. That represents a lot of potential energy, right? So you have this big, large amount of water behind the dam wall, and then what? A small amount of water, like a much smaller version of that river, trickling away at the bottom. So how do you harvest that potential energy to do things like make electricity? Well, you put little holes in the wall that you control the flow of water passing through, and where the water passes through, you have something called a turbine, like a little paddle wheel. If you've ever seen an old paddle wheel dam, where the way people used to grind flour, they would harvest a little energy from rivers. You have this spinning structure with little paddles on it. As water passes over that spinning structure, right, it's turning that little paddle wheel, and that kinetic energy can now be used to, in our case of a hydroelectric dam, make generate electricity, convert that energy to electricity. In the case of electron transport in the mitochondrion, what you're doing is you're making a big hydrogen ion gradient. So instead of piling up a lot of water on one side of a wall, you're piling up hydrogen protons. Now, if you're moving against the concentration gradient, if you're moving things from lower concentration to higher concentration, that requires active transport. Where's that energy for the active transport to keep shoveling those hydrogen protons across the membrane to make that gradient? The energy from those electrons. As I said, each time the electrons get transferred, they give up a little energy each time. That little energy is being used to push hydrogen protons across the membrane, building up that big gradient that is eventually going to pass through the ATP synthase protein. Remember in the video you saw that little turbine, that little spinning turbine? That's ATP synthase. And so the only way those hydrogen ions can cross that membrane, because you've got a lot of potential energy. Think about this. You've got a lot of hydrogen protons on one side, not too many on the other side. If you let them through, they're going to zoom through there. The only way they can cross is through ATP synthase. As they pass through, as they are zooming back to the other side, it spins the turbine. That energy is used to attach that third phosphate onto ADP, forming ATP. Now, if you think about it, it's like, wait, if in a river, in a hydroelectric dam, the river keeps taking water away, right? It doesn't just pile up until you have an even amount of water on each side of the wall. So how do you get rid of some of those hydrogen protons so you don't have an even number on each side of the membrane? That's where oxygen comes in. So a lot of those hydrogen protons crossing that membrane, if you just let them pile up, eventually you'd have no gradient, right? You'd have an equal concentration on each side of the membrane. So you got to do is get rid of some of those hydrogen protons. And the way you do that is you bind them up with those used electrons to oxygen, forming water. So mitochondria have structures that act like turbines. Each one of these tiny machines is called ATP synthase. So it's an enzyme, right? It ends in ASE. And since the name usually tells you what's going on, this is an enzyme that synthesizes ATP. So these proteins are built into the inner mitochondrial membrane, adjacent to the ones that are doing the passing along of those electrons. And here we see what's happening. So again, keep in mind, this is the inner mitochondrial membrane. So if you look on the right-hand side of the diagram here, you see those little inner folds in the mitochondrion. Embedded in those membranes are these proteins. So what you're doing is, every time you're passing those electrons, you see those electrons being given up at the left-hand side of the diagram, right? NADH at the first protein, FADH a little later on. So in the beginning of this chain, the electron carriers give up those electrons. Those electrons get passed from protein to protein. Each time they do, they lose a little energy. What's that energy doing? Pumping hydrogen protons against the concentration gradient, right? So if you're going to go against the concentration gradient, you need to use energy. That energy from those electrons is being used to push hydrogen protons against the concentration gradient, yielding a big gradient, right? So a lot of hydrogen protons in that space between the membranes, relatively few, in the matrix on the other side of the inner membrane. So the electrons get passed along, each time losing energy. What's happening with that energy they're giving up? They're pumping hydrogen protons across that membrane. At the very end, okay, you got spent electrons, and you got to clear out some of those hydrogen protons to keep that gradient, pretty high. So what happens? Oxygen comes along. Oxygen mops up. So one half of an O2 molecule grabs up those electrons, scarfs up a couple of hydrogen protons, and forms water. So how do you harvest that big concentration gradient of protons? It's that last molecule to the right-hand side. That's the ATP synthase. And that's the only enzyme you have to know in cellular respiration in this course. And what's happening is it's allowing the hydrogen protons to now move along the concentration gradient. Passing through, the energy spins. It's weird. It's like you usually don't see things that spin like this in nature. If you think of like a car wheel or a wheel on an axis. But this little guy spins. And as it spins, that energy from spinning is used to stick that third phosphate onto ADP, forming ATP. And that's where the big payoff is for your glucose molecule. Yeah, you make a couple at glycolysis, you make a couple at citric acid, but the largest amount of ATP is right there, ATP synthase. So it's not even being made along electron transport, it's only at ATP synthase where you actually synthesize most of the ATP. So this is just the same diagram, just a little closer. So electron transport is passing electrons along this little bucket brigade. Each time a little bit of energy is lost, that energy is being used to pump hydrogen protons. So now you've got a big concentration gradient. That represents potential energy. How do you harvest that energy? You let those protons pass through that one point, that enzyme, which then converts that energy of the concentration gradient to energy attached to, to join a phosphate group to ADP, forming ATP. Okay, so this gradient is how you generate that big hydrogen proton. These proteins are how you generate that big hydrogen proton gradient. This is where it actually gets used. So that's where the energy that was harvested during all those reactions along the way, during glycolysis, during citric acid cycle, all the energy represented in all those electrons was used to make that gradient, and then that energy is now being harvested to form ATP. So if you look at a hydroelectric dam, it's kind of like that. If you think of each of the openings in that dam representing an ATP synthase molecule, that's essentially what's going on. You have turbines in the dam that's taking that energy and converting it to electricity. Well, you're converting electron energy that's represented in the proton gradient instead of a water gradient, and using that energy to make ATP. Okay, so skipping over this. So, cellular respiration can generate up to 32 molecules of ATP per molecule of glucose. So here's what I call the accounting part of the program here. So, I like this diagram because it again reminds you, glycolysis happens outside the mitochondria. The other reactions occur inside the mitochondria. So the ones inside the mitochondria are the ones that require oxygen. The reaction out here in the cytoplasm does not. So you split glucose to make 2-pyruvic acid, right? That's glycolysis. You spend 2 ATP, you make 4 ATP, so you have a net gain of 2 ATP by what they call direct synthesis, right? So actually sticking that phosphate onto the adenosine diphosphate. You've captured some energy in the form of high energy, you've captured some electrons by the electron carriers, in this case 2 NADH. So those are going to enter into the mitochondria, and they're going to give up those electrons at the electron transport chain. Okay, so your pyruvic acid are entering into the mitochondrion. You have that series of steps that generate the 2 acetyl-CoA molecules per original glucose. At that step, you've generated 2 more NADH, right? And those are going to give up their electrons at electron transport chain. The acetyl-CoA then hops on the merry-go-round of doom and gets broken the rest of the way down. So by the time you're done, you gave up 2 carbon dioxides at this acetyl-CoA step. You give up 4 more carbon dioxides during citric acid. So there's all 6 of your carbons leaving the reaction as carbon dioxide. You've now finished breaking down that original 6-carbon glucose molecule. And during these steps, you load up a whole bunch more electron carriers, right? Because you're harvesting all that energy that was originally holding together a glucose molecule. As you break it down, you then take that energy and you basically save it in the form of these electrons, loading up these electron carriers. during citric acid, you yield 2 ATP by direct synthesis, right? So you get a little bit glycolysis, a little bit in citric acid. All those electron carriers are now going to the electron transport chain. They get passed along those proteins. That little bit of energy being given up with each step is used to make that big hydrogen proton gradient, which is that energy is now harvested when those protons rush through ATP synthase. As it spins, that energy is now attaching that third phosphate to ADP, forming ATP, and there you yield about 28 ATP. So obviously a big harvest versus what you saw in the earlier steps. So 2 at glycolysis plus 2 at citric acid plus 28 at electron transport is about 32 ATP. And the reason why you see about at those, referring to electron transport chain, is that it's not perfect, right? It would be awesome if the membranes all perform perfectly and all that, but it's never. It's like there's a little leakage here and there, so it's not a perfect conversion of energy, and that's why you see the about. It's not as exact, because there are some inefficiencies in that membrane. So, viewing this outside of the context of the mitochondria. So this is ATP yield during cellular respiration. So again, big payoff is at electron transport chain. Now, I keep saying glucose when we talk about these processes. Keep in mind that respiration is a versatile metabolic furnace, right? So think of a furnace that instead of just burning wood, you can throw in paper and you can throw in other stuff to burn to warm your house. This furnace can burn other types of food molecules. So the next diagram is going to show you some metabolic routes for other carbohydrates, fats, proteins to be used as fuel for cellular respiration. So keep in mind that it isn't that everything has to be turned into glucose to be used in respiration. If you think about how starvation works, right? Probably a kinder way of thinking about it. If you think about someone who goes on a low carb diet, right? Eventually you burn through the carbohydrates because you're not ingesting very many. So what happens? Well, the body then turns to breaking down fats for energy. The downside is if you take it too far, eventually you burn through the carbohydrates, you burn through the fats, and then your body is like, okay, I got to start breaking down proteins. Now, at a low level, that's not so bad, but if someone is really, truly starving themselves, eventually the body starts attacking its own proteins as fuel. And you start seeing things that are often associated with disorders like anorexia, where there's heart damage, right? Where the body is basically breaking down proteins in muscle to fuel respiration. So there's moderating one's weight, and then there's taking it way too far. So, using this delicious little sushi roll as an example, all right, that's the food. So let's say we look at carbohydrates. Yeah, there are things like sugars, but there's starches, right? You break down the starch in the rice, eventually breaking it down to sugars. Okay, sugars can go into glycolysis, just as we have already talked about in this lecture. What about lipids? Well, fats can get broken down into glycerol and fatty acids. The glycerol can enter the glycolysis stage and go through the rest of the steps. Fatty acids can actually basically jump right in at the acetyl-CoA stage. Proteins get broken down to amino acids, which can enter at all 3 points. There's different ways in which amino acids get used, but they can kind of jump in at all 3 stages along the way. But ultimately, you're going to be breaking these guys down, harvesting electrons, using those electrons to power the electron transport chain, which ultimately gets you the ATP. So yeah, there's other ways to get energy out of food. So popping over that. Now, remember back in the beginning I talked about anaerobic respiration. You're running for the bus and eventually get to the point where you're running more than your breathing can bring oxygen to your muscles. So you can do this. Your cells can work for short periods without it. So this anaerobic pathway without oxygen is called fermentation. Fermentation relies on glycolysis, the first stage of cellular respiration, because as you recall, that takes place in the cytoplasm. It doesn't require oxygen, but you do get 2 ATP molecules for each glucose molecule broken down to pyruvic acid. So instead of getting a number up in the 30s, you're getting 2. So it's wicked inefficient. Better than nothing, but for a big organism like us, not a whole lot better. So it's basically an emergency pathway. It's inefficient as get out. You wind up with these big waste product molecules, but it's better than, again, nothing. So to harvest food energy during glycolysis, NAD+ must be present to grab electrons, right? That's your first electron carrier. And that's cool under aerobic conditions because the cell regenerates NAD+ every time NADH is dropping off the electrons over an electron transport chain. This recycling of NAD+ can't occur under anaerobic conditions because there's no O2 to ultimately accept the electrons. Instead, NADH gets rid of its electrons by adding them to the pyruvic acid produced by glycolysis. So what do you form by this? A waste product called lactic acid. So I'm going to skip to the smaller diagram here. So input is glucose, just as we've seen before. Okay, glycolysis, all right, we split the, we harvest some energy in electron carriers and we generate a couple of ATP. But how do you reset it? Well, it transfers those hydrogens onto the pyruvic acid. What you wind up with is lactic acid. So the waste product is big, right? You're not breaking that 6-carbon glucose all the way down to 6-carbon dioxide and all that energy got scarfed up by electron carriers. You've got 2 big lactic acid molecules, so most of that energy is still bound up there. You've only harvested a little bit in ATP. So what goes in is glucose. What comes out is 2 big old lactic acid molecules. So a lot of that energy is still bound up in those chemical bonds. So again, what do you do? How do you unload your electron carriers? You dump those hydrogen protons onto the pyruvic acid. You've yielded the net 2 ATP, but now you got to get rid of the electron. So slaps it onto pyruvic acid, form that lactic acid, and that can build up and eventually leads to discomfort if you've ever worked out past your aerobic capacity and you get the ow.
All cells are able to synthesize ATP via the process of glycolysis. In many cells, if oxygen is not present, pyruvate is metabolized in a process called fermentation. By oxidizing the NADH produced in glycolysis, fermentation regenerates NAD+, which can take part in glycolysis once again to produce more ATP. The net energy gain in fermentation is 2 ATP molecules per molecule of glucose. Fermentation complements glycolysis and makes it possible for ATP to be continually produced in the absence of oxygen. There are two types of fermentation. Alcohol fermentation, which occurs in yeast, results in the production of ethanol and carbon dioxide. Lactic acid fermentation, which occurs in muscle, results in the production of lactate.
Right, so again, because this is happening only in the cytoplasm, you don't get, this whole business of going into the mitochondria, you don't get to break it down all the way down and harvest all that energy. Now, what causes muscle burn, right? The old expression, feel the burn in people exercising, or why? Right? What causes muscles to feel that burn after hard exercise? Well, this was investigated by one of the people who made the field of exercise science, a fellow named A.V. Hill. He actually got the 1922 Nobel Prize for investigating muscle contraction. So think about this. It was only 100 years ago, which may say yeah, that's 100 years ago. But if you think about it in terms of how long people have been understanding the world, this is only about 100 years ago that this was even worked out. Anyway, he knew that muscles produce lactic acid under anaerobic conditions. So what he did was he developed a way to electrically stimulate dissected frog muscles in a laboratory solution. So they unfortunately killed frogs. they took the freshly killed frogs, typically the big leg muscles, and then attached them to a series of sensors. So what's happening here is these are two, again, big, the big thigh muscles off of frogs. On the left-hand side, you have battery, right? This is energy coming in, and that electricity would stimulate the frog muscle to contract. And so you have a tension meter, basically this little structure that measures when that muscle contracts, it measures that force generated by the muscle contracting. Now, the first solution on the left prevents diffusion of lactic acid. On the right-hand side, you have the same setup, but this solution allows diffusion of lactic acid, and the muscle can work for twice as long. Right? So as long as you get rid of that waste product, it can keep going for much longer. So he showed that when lactic acid cannot diffuse from muscle tissue, muscle performance declines. When lactic acid could diffuse away from the muscle tissue, performance improves significantly. And you might say, awesome, we figured this out. Science sometimes will just contradict all your good work. Eventually, evidence began to build up that contradicted his results. For one thing, the effect he demonstrated did not appear to occur at human body temperature. He was using frog tissue. Frogs are ectotherms. They're not warm from metabolism like we are. So he was trying to see, like, okay, let's see what happens in humans. He didn't seem to get that same effect at human body temperature. Further, some people are unable to accumulate lactic acid. They have muscles that fatigue faster, right? If you can't build up lactic acid, you'd think, oh, your muscles would never tire. But for some reason, they tire faster. So that's completely opposite of what you would predict. So there's research that's looking at other things. They think that increased levels of certain ions might be to blame. So we know some of it, but the real true cause of muscle fatigue is something that people are still working on and arguing over to great degree. Now, some things on this earth live with or without oxygen, one of those being yeast. In the lab experiment, you put yeast in a sealed container. So they'll burn through the oxygen in that container. Once that's gone, they switch from the aerobic respiration using oxygen to the anaerobic process of fermentation. Waste from this type of fermentation can be ethyl alcohol plus carbon dioxide versus including also the lactic acid that we saw in the previous example, or other compounds. It depends on the type of yeast. There are many different types of yeast. So depending on what type you're using, you can get lactic acid as a byproduct. Think of fermenting stuff like cabbage to make sauerkraut. If you want to raise bread or make things like beer, you want to use a type that will have a waste product of ethyl alcohol. All right, so depending on what type of yeast you're using and what type of substrate of food you're giving them, you can get different products. But yeasts are what they call facultative aerobic organisms. So they can live aerobically, they can harvest a lot more energy aerobically, like any other organism, but they can still survive under living just anaerobically. So let's look at the ethyl alcohol type of fermentation. So again, input is glucose. You go through glycolysis, right? So you get that 2 net ATP. You load up the electron carriers, but get a slightly different next step. You take those 2 pyruvic acids and you release those carbons in the form of carbon dioxide. And there's multiple steps in here. I'm trying to keep it fairly simple. You basically unload the electron carriers. They bind up some of the electrons are basically dumped off into hydrogen. You wind up, instead of having pyruvic acid picking up picking up additional parts to assemble that big lactic acid. Here you do shave off 2 carbon dioxide per glucose, so your output is not quite as huge. It's this ethyl alcohol molecule, so it's a little bit more efficient than lactic acid fermentation. So instead of input being glucose and the output being 2 lactic acid, we have input being glucose and the output being 2 ethyl alcohol molecules. Right, so a slightly different way of going about it. But both are fermentation, both are anaerobic means of harvesting energy, and both are pretty inefficient per glucose molecule. So, aerobic and anaerobic respiration both start with glycolysis. So this glycolysis is actually the universal energy harvesting process of life. The role of glycolysis in respiration and fermentation actually has an evolutionary basis. If you go back to the early Earth, as I mentioned in the last section, between 3.5 and 2.7 billion years ago, there wasn't a lot of oxygen in the atmosphere. So if you think about it, if there wasn't a lot of oxygen around, the ancient prokaryotes probably used glycolysis to make ATP and generated ATP only from glycolysis. You might say, well, you don't get a lot of energy. Well, if you're only a single-celled organism, you don't need a wicked amount of energy to pull off life. So it's kind of interesting where the earliest life was definitely being anaerobic. Once the autotrophs started producing oxygen, accumulating oxygen in the atmosphere, once it hit about 10% of modern levels, we see the first eukaryotic organisms show up. So there's probably an association with making a more efficient way of harvesting energy with larger, more complex, much more complicated cells like we see in eukaryotes. Okay, so, and again, you can see, this reddishness. If you've had metal on a car exposed to moisture and water, you get rust, right? You've seen rust on cars. Well, you can see rust in geologic formations, right? The oxidation of materials, and that's part of our evidence as looking at the layers of land, seeing which layers start developing that sort of rusting going on. So last thought in the chapter, the fact that glycolysis occurs in almost all organisms suggests that it evolved very early in ancestors common to all the domains of life, right? Both prokaryotic and eukaryotic. The location in the cell also implies great antiquity, right? It happens in the cytoplasm, not in a membrane-enclosed organelle, right? So this is taking place in a fluid that is present in all cells, right? It doesn't require that membrane-enclosed organelle. It probably evolved more than 1 billion years after the prokaryotic cell. So glycolysis is something we've inherited from our most early ancestors of living things on Earth. So with that, go enjoy a snack and think about how your body's processed it.